So the previous speaker set up very nicely how the proton gradient is related to ATP synthesis. But I also have a little bit of an introduction on that, which I'll go through faster now thanks to his presentation. But just to get us all on the same page, I had these first two slides. So basically, metabolism consists of two parts. The part where you build up complex organic molecules from simpler ones, that's called anabolism, and that requires energy, which is obtained by converting ATP to ADP and oxidizing NADH to NAD+. So that's shown in this arm of the schematic diagram here. And the other part of metabolism is the breakdown of the complex molecules, which releases energy, and that's called catabolism. And that released energy is then stored in the formation of ATP and NADH, and that's shown in this arm of the cycle on this schematic. The abbreviations are shown over here. So how is this ATP synthesized? ATP then is a very key molecule for all metabolism, and so are these so-called cofactors such as NAD. There are various ways of synthesizing ATP, but in most, depending on the metabolism of the organism. So in most organisms, except for the fermenters, ATP synthesis depends on the formation of a pH gradient that's called the chemiosmotic potential across the cell membrane. And coupled to the generation of this pH gradient is the generation of NADH from NAD. And this entire process is achieved by transferring electrons down an electron transport chain that involves enzymes. I'll go through the schematic in a minute, but let me just finish reading these bullet points. The movement of protons down the chemiosmotic potential drives the synthesis of ATP by this ATP synthase molecule that was presented in the previous presentation as well. The enzymes that are involved in establishing the chemiosmotic potential contain these iron-sulfur clusters, and the subscript N simply means that this is the stoichiometry of the iron and sulfur. So they contain these iron-sulfur clusters at their active sites, and it's been therefore proposed for a long time in the literature that iron-sulfur minerals might have played the roles of these enzymes in protocells. So what are we talking about over here? Here's the membrane, the phospholipid membrane shown here in the background. This represents the interior of the bacterial cytoplasm, and that's the outside or the periplasm. And in the case of this picture is taken for photosynthetic metabolism, the electron transport chain. So you have photosystem 2, where the photolysis of water produces, not photolysis of water, but where water is broken down to form oxygen and 2 protons. So this generates protons out here. And the electrons in the process are picked up and transferred across to the cytochrome complex here by a quinone, plastoquinone. And the plastoquinone and the cytochrome complex spit out more protons out here. Further transfer the electron using plastocyanin to photosystem 1. Photosystem 1 transfers it using ferredoxin to NADP reductase. And at NADP reductase, a proton is combined with NAD to convert it to NADH. And so a proton is consumed over here. So protons are produced on this side and consumed on this side, and this generates a net enrichment of protons on this side of the membrane, a net depletion of protons on this side of the membrane. So this creates this chemiosmotic potential, and the fact that these electrons are being transferred from one enzyme to another, this is the electron transport chain, and here are the iron sulfide clusters that are in the active sites of all these different electron transport enzymes. Here's another picture now, continuing on. Once you've generated this chemiosmotic potential of more protons on one side than on the other side, they flow back through the ATP synthase, where ADP combines with phosphorus and is converted into ATP. So the question was posed in the previous talk, and we will talk about it here as well, as to how did this chemiosmotic potential first arise in order for ATP synthesis to occur. So the hypotheses that we have built in our model, in our experiment here, are based on a couple, the experimental system we built here are based on a couple of hypotheses. The first hypothesis is the earliest protocell metabolism was fermentative. This was proposed by Lazcano and Miller in the 90s. And the idea is that those organisms didn't have this proton gradient to drive ATP synthesis. But at some point, there was some evolutionary transition to metabolisms that did have the electron transport chain and chemiosmotic potential in order to drive ATP synthesis. So the hypothesis we have in our work here is that photocatalytic minerals may have played the roles of enzymes in this evolutionary transition, including iron-sulfur minerals, which are such as pyrite, which are photocatalytic. So we're going to build an experimental system to test this hypothesis, whether photocatalytic minerals can actually drive this NAD to NADH reduction reaction, as well as create a transmembrane chemiosmotic proton gradient. So we've devised a model protocell consisting of a lipid membrane, the lipid vesicle bilayer. We're going to react we're going to use a photocatalytic mineral and shine UV light on it, which generates a hole and an electron, since it is a photocatalytic mineral. On the outside of this membrane, we have a reductant. We are choosing any common reductant that might have been present in the environment, such as an amino acid. We just picked serine. It can be any amino acid or any simple organic compound. The serine takes up the hole and combines with water to produce glyoxylic acid, and it also produces protons in the process. Meanwhile, the electron is picked up by a molecule called a polyaromatic hydrocarbon, which was included in the membrane during the synthesis of this protocell vesicle. So the polyaromatic hydrocarbons have been shown previously to be electron shuttles, and their structure is actually very similar to the quinones that we saw that act as electron shuttles in the actual biological system. So in the step 1 of this electron transport chain, the electron is picked up from the photocatalytic mineral transferred to the polyaromatic hydrocarbon. In step 2, the polyaromatic hydrocarbon picks up the electron, passes it through the membrane to an electron mediator. In our case, this is a model compound called rhodium bipyridinium, which the structure is shown over here. Now, this is not very prebiotic. Rhodium is not very common in the environment, but we use this because it's capable of a 2-electron transfer, which is what you need for NAD to NADH reduction. We are currently in our lab trying to also work now with the iron analog of this, which would be more prebiotically plausible. And finally, in step 3 of this process, so when this rhodium bipyridinium picks up an electron from PAH, it's reduced to the +1 form from a +3 starting out form. And this reduced molecule then reduces NAD, the cofactor which is included in this vesicle, and reduces the NAD to NADH. In the process, it itself gets oxidized back to the 3+ form. NAD is prebiotic. It has been synthesized by Steve Benner's group in a prebiotic chemistry recently last year. And the structure of NAD and NADH again are shown over here. So this is where the H is added to make it an NADH compound. So we have built this entire system in the lab, and we follow the reaction, whether it's actually happening, by monitoring the production of NADH over time by using fluorescence spectroscopy. How much time do I have? Okay. So what we're going to do now is going to look at a lipid vesicle with various different minerals. We tested various photocatalytic minerals using a serine reductant, and we're going to look at the production of NADH when UV light is shown on the system. So here are the results. Here is the fluorescence as a function of the scanning wavelength. And you can see that at a certain wavelength, there is a maximum in the peak, which tells us that NADH is being produced. So after at various time points of 1 hour, 2 hours, and 3 hours of irradiation of UV light, you can see that the peak is increasing. That means NADH production is increasing in our protocell system. So we achieved this using pyrite and greenockite, and we also use cadmium selenide. Again, this is not prebiotic, but this is just as a case study. Cadmium selenide are quantum dots, and they're known to have photocatalytic activity. Titania is also photocatalytic and produces this NADH signal beautifully, as well as zincite. We also tried sphalerite and chalcocite, but they didn't work. And it might be that these were all synthetic minerals that we bought, so their particle size is really small, and it's in the right particle size to be catalytic. These ones were natural minerals, and we had to crush them up, and we probably didn't get down to you can't crush things down to the nanometer size. You can only get like 100, 200 micron size particles, which may not be in the right size range to be photocatalytic. So we didn't get the reaction with these sulfide minerals. Now, so anyway, there's a range of different minerals which are showing this property of NADH production. The next thing we wanted to test is this is just a bar chart that quantifies the amount of NADH produced as a function of time, and you can see that titania is the most efficient, and then followed by cadmium sulfide, zinc oxide, cadmium selenide, and the least efficient actually is pyrite, but that doesn't matter. There was plenty of pyrite around on early Earth, and by 3 hours, it's doing a pretty decent job of producing the NADH. Now, that was using a lipid membrane which was based on phospholipids, and phospholipids have actually been synthesized in prebiotic syntheses, so it's not as prebiotically implausible to use a phospholipid membrane as some might think. But just for our satisfaction, we also tested this with a fatty acid membrane. So we used oleic acid, which is a commonly used fatty acid and is considered to be more prebiotically plausible than a phospholipid, and we are able to produce NADH in exactly the same way using the same system setup with serine as the extravesicular or extracellular reductant, terminal electron donor. So this compares the oleic acid system to the phospholipid system that we just looked at in the previous slide using serine, but we also decided to change up the extracellular reductant and use 3 different ones. We've used serine, we've used glycine, and we've used isopropanol, and as you can see, pretty much any simple organic reductant can work in this reaction scheme, and we get the production of NADH. Ignore these for the moment. I won't get into the details of this. If anybody has questions about these, I can come back to them. Basically, these graphs show that the NADH being produced is the biologically active isomer, which is the 1,6 form of NADH, and not the inactive form, which is the 2,3 version of NADH. So we were able to generate this protocell drive the reduction of NAD to NADH using a photocatalytic mineral with different lipid membranes and with different reductants. So the system is quite robust. What about the generation of the transmembrane pH gradient? What we do is we enclose this fluorescent molecule inside the vesicle, and this fluorescent molecule is sensitive to changes in pH. Over a period of 3 hours, as we irradiated our system, and as the NAD was being reduced to NADH, we saw that the fluorescence intensity inside these vesicles was changing, and by calibrating it to a pH curve, we were able to determine what was the pH inside the pH inside the vesicle. So at the starting point, at time 0 hours, the pH is the same as 8.6, which is the pH at which the vesicles were synthesized. So the outside and the inside are both at pH 8.6. When we use the phospholipid, which is a very impermeable membrane, the pH doesn't really change much over time. Thank you. When we use the oleic acid phospholipid mixture in a ratio of 5:1, the pH drops over a period of time, and this is not surprising because oleic acid vesicles, fatty acid vesicles, are not as impermeable. So what's happening is that the protons are getting carried in from the outside, where the protons were generated by, remember the serine, when it is oxidized, it produces the protons. So the outside of the vesicle is getting acidified, and that pH gradient is being maintained in the case of the POPC vesicles, but it dissipates over a period of 3 hours because the fatty acids shown by these orange molecules here do this lipid flip-flop. So they're initially deprotonated. As the exterior becomes more and more acidic, they pick up a proton, and they do the lipid flip-flop and carry the the proton from the outer leaflet into the inner leaflet and release it into the inside of the vesicle. And so that's why the interior starts getting acidified. But basically, by the time the evolution of phospholipid membranes had come about in these protocells, you would have had a pretty tight membrane which was capable of maintaining this chemiosmotic potential. So in conclusion, various photocatalytic minerals are capable of promoting a transmembrane electron transfer chain with reduction of NAD plus to NADH and the generation of a transmembrane pH gradient. These reactions utilize an extracellular organic compound as the terminal electron donor, such as serine and glycine. Thus, the model represents a proto-photoheterotrophic metabolism because it's utilizing this reduced organic compound for the metabolism. The minerals we are showing actually, there are lots of hypotheses that minerals may have played the roles of enzymes in protometabolism, but this is one of the first cases where there's a demonstration that that's actually working. And it's happening in a photoheterotrophic metabolism. As membranes evolve towards more phospholipid-rich compositions, this developed chemiosmotic potential would have occurred, and as evolution proceeded, this might have eventually been used for the synthesis of ATP as the evolution eventually produced an ATP synthase. So I'd like to conclude with thanking my postdoc, Poonam Dalai, who did all of this work, and was assisted with a really amazing graduate student, Puthu Ustriyana, and funding from the Simons Foundation, National Science Foundation grant, University of Akron startup funds, and very generous gift funds from a person who's just interested in origins of life research. And thank you for your attention.
Thank you very much. Unfortunately, we don't have time for questions as the session has concluded. I'd like you to, let's all join together and thank all of the speakers from the session.